Welcome to Section 1, Using the BLAST tool to analyze a DNA sequence. By the end of this section, you will be able to demonstrate the skill for applying a technology, in this case the BLAST tool, for DNA data analysis. Before we move on to the first topic in this section, you need to refresh your knowledge on two important tools we have mentioned before. The first one is about the BioEdit tool. This is one of the most popular software used to view or edit sequence files. These sequence files can be DNA, RNA, or even protein sequences. There are many other functions of BioEdit which we will be discussing about in the future sections of this course. In Section 1, we will be using a DNA sequence. The next important tool is the NCBI website and its functions. Right throughout this course, you will need to access the information, obtain data from the database, and use the many features or tools available on this website. In this section, however, we will be focusing on using the BLAST tool. So, let's start learning the basic steps you need to follow to use the BLAST tool and analyze a DNA sequence. Section 1.1 Checking the quality of a DNA sequence. The objective of this section is to help you identify the features of a good quality sequence. Before we start any analysis, we need to confirm that the quality of our DNA sequence file is good. This is very important because if you use a sequence file with low quality, it could lead to faulty analysis or incorrect results. So, how do we check the quality of our DNA sequence file? The first step that you could do is to open your sequence file using BioEdit. To do this, first open the BioEdit software in your computer. You will see a window like this. To open a sequence file, you can click on the yellow file icon at the top left-hand corner or go to File and click Open in the drop-down menu. Once you click Open or on the yellow icon, you will get a pop-up window. In this, go to the correct folder and select the sequence file you need to open. Do you see that the sequence files are in AB1 format? This is the format in which you will receive your sequence file from the sequencing company. It would be better if you save your sequence files, especially those related to one analysis, in one folder on your computer. Then it will be easy to access them. After you select the correct file, click Open. Once you click Open, you will see two files like this. In one, you will notice the DNA sequence in letters. This is the sequence of the 4 nitrogenous bases of DNA. A for adenine, G for guanine, C for cytosine, and T for thymine. Now look at the other file. Here you can see a sort of graph. We call this a chromatogram. Each nitrogenous base is represented by a color, and each peak corresponds to one of the 4 bases. You can use the vertical scale and horizontal scale to increase the height or width of the peaks. To determine whether this sequence is good or bad, we must check the peaks throughout the chromatogram from beginning to the end. If, for most of the chromatogram, the peaks are neat and in order, and there are no double peaks or 2 peaks at once, then the sequence is a good sequence. Most of the time, at the beginning of a sequence and at the end of a sequence, you will not find neat peaks. This is quite normal, and we can omit these parts when we do our analysis. However, if you observe something like this, which we refer to as noise, throughout the whole chromatogram, that means your sequence is of poor or low quality. Therefore, you should be cautious in using such sequences in your analysis. Now we have come to the end of section 1.1. So far, we have discussed the importance of having a good quality sequence, because a bad quality sequence will lead to incorrect results. So to determine the quality of your sequence, you need to check for neat peaks in the DNA sequence before you use it for any analysis. Here is a small question for you. See if you can answer it correctly. Yes, this is a chromatogram. You can also attempt this question for homework. In this section, we used BioEdit to open and check the quality of a DNA sequence. Can you find some other tools we can use instead of BioEdit? I will be discussing some of them in the subsequent sections. Let's discuss then. Thank you.